So thank you all for joining today. We are going to hear from Garrett McKinney, who is a relatively new member of WDFW. He started mid-pandemic, so unfortunately we have not had an opportunity to welcome him in person, most of us, but being a geneticist, at least he has more access to the building than some of us do. So he joined in December 2020. He did postdoctoral research at the UW and at NOAA. And his recent work includes development of marker panels, which probably some of us know about and hopefully we'll learn a little bit more about, and but focused on Chinook. And so today he will talk about adaptive markers and how and have a management focus, which is pretty cool given that we are a management organization. Hopefully can translate some of the genetic speak to some more real world applications. So Garrett, thank you so much for volunteering to speak, and take it away.
Nope. Okay, I'm gonna turn off my camera just to try to save some bandwidth if I don't know if that actually is going to make a difference. So yeah, thanks for having me. I'm Garrett McKinney with the Molecular Genetics Lab. Started back in December, and today I'm going to be talking about how to bring adaptive markers into management. So right now, genetic markers are typically broken down into two categories, either neutral or adaptive. Neutral markers are typically assumed to have no influence on phenotypic traits or fitness. And so this cartoon example on the right side, can you see my cursor?
I don't think so. Maybe there's
Okay.
a way to turn it on. If anybody else knows, maybe you could write it in the chat.
Yeah,
I can see it.
Okay.
Oh.
Yeah, it's handy to know if people can tell what I'm pointing at or not. So these two fish on the right have different genotypes. One has an AA, one has a TT, but it doesn't have any influence on their ultimate phenotypes or their fitness. They're identical for all intents and purposes, even though those genotypes differ. In contrast, adaptive markers are assumed to directly influence phenotype through their effect on directly influence fitness through their effect on important phenotypes. So the example below, we have the AA fish is much smaller than the TT fish because of that particular genotype. And as a result, there's a fitness difference between them because of different body sizes. Traditionally, management has focused on using neutral markers to discriminate among populations. And in general, this works well, but adaptive markers can give us more information. And one of the things that we can do with that is we can use them to get information about important phenotypic traits that might be impossible to measure at some life stages, such as likely maturation age or return time for juvenile fish. So knowing the ultimate age of maturation could be useful for things like stock assessment, but if you catch a juvenile fish in the ocean, you don't know how old it's going to be when it finally matures. But if we have markers that are associated with age at maturity, we could probably assign a predicted age at maturity or a likelihood of a given maturation age. And that could give us more information that's beneficial for management. With return time, there's a number of populations where at neutral markers, we can't tell fall or spring fish apart. But with adaptive markers, we'd be able to tell if that was a fall or a spring fish. Neutral and adaptive markers also don't always show the same pattern of population differentiation. There's lots of cases where if you look at the neutral markers, it'll look like a single interbreeding population, but ecotypes would show differentiation at adaptive markers. So these figures on the right are from a paper by Larson et al. Looking at sockeye salmon in the Wood River. And there were 3 different ecotypes that were examined, beach spawners, river spawners, and stream spawners. And when they looked at populations with these different ecotypes, when you look at neutral markers, the beach and the river ecotypes look similar to each other, and the 2 stream ecotypes are diverged from each other and from the beach and the river fish. Presumably, this is because of genetic drift causing differences among the populations. But when you look at markers that are in islands of divergence in the genomes, which are putatively adaptive markers, we see that the samples instead cluster by ecotype. So these give us different levels of information. One just gives us the neutral population structure, and the other gives us differentiation by ecotype. So with these added markers, we can tell the beach and the river fish apart where we couldn't before. So with the genomics revolution, it's been a really exciting time to be in science. The next generation sequencing technologies that are out allow rapid and inexpensive sequencing of millions of small DNA fragments. And in practical terms, this means that we can have whole genomes sequenced in a single day, or multiple whole genomes, depending on the equipment being used. And on the other end of the scale, we can genotype fish for hundreds of genetic markers, but genotype thousands of them at a time. And this low cost of whole genome level sequence has allowed researchers to better identify genetic variants that are associated with phenotypic traits. One of the caveats with that is that initially sample sizes are often small, and so you don't have the power to detect all genetic variants. What often is detected first are the genetic variants that have a large influence on the trait. But also a benefit for us is those are the most readily used for conservation and management, because it's much easier to try to interpret one or a few markers instead of trying to predict a phenotype from hundreds of different genetic markers. And this sort of relates back to how the ultimate genetic control of traits. A common assumption is that most traits are under the control of many, many genes, each having a small effect. And this is often true, but we're finding increasing examples where one or a few genes do have a large control over a trait. And so that's the sort of example that I'll be focusing on for this talk. And right now, we're really just at the tip of the iceberg for identifying genetic variants that influence phenotype. I'm sure there's going to be many more examples out in the coming years as more and more researchers are doing whole genome sequencing. So historically, the process of identifying adaptive genetic variants took place in multiple steps. And a lot of times this is still what's going on. These figures here are from a paper by Johnston et al. Looking at horn morphology in soy sheep. And this is sort of a good example of the basic path that is often followed. They started off by looking at 50,000 markers throughout the genome. And so there's a lot of genetic variants that they're looking at, but it's not the whole genome. There's a lot of spots where they just don't know what's going on. They were able to find a high association with horn morphology on chromosome 10, and they were even able to identify a gene that was likely associated because it had both had the peak association and the function of that gene made sense for influencing horn morphology. But even though they knew this, they didn't know which specific genetic variants in that gene were influencing the phenotype. So the next step is to do more sequencing within this region of interest, identify genetic variants that have a high association within that region, and then genotype more individuals for those specific variants to confirm that there's an effect on the phenotype. And in this case, you can see that there's a pretty strong effect on horn size and horn circumference based on the individual's different genotype. One thing that will start happening more often as people are able to do whole genome studies on a large number of samples at once is that a lot of these initial steps will be able to be skipped, and the whole genome, a single whole genome analysis will be able to get you all of these results. So I'm going to focus on adaptive markers in salmonids, primarily because most of my work is on salmon, and that's just what I'm familiar with. But certainly these things are going on in other species as well. In salmon, genetic variants have been identified for some key traits. These include age and size and maturity, migration versus residency, and run timing. And I listed some of the species that have these associations. And in this talk, I'm going to focus on cases where we've identified specific genetic variants associated with the traits rather than just genomic regions. There's certainly a lot more studies that have identified genomic regions associated with traits, but unless we have specific variants that have a high association, then there's not a lot that we can currently do with our marker panels. So I'm going to go into a bit of detail for each of these just to show you sort of what's known with these different traits and where we're at with the state of knowledge. So age and maturity it's obviously an important trait in salmon. It's associated with reproductive success, where larger, older individuals tend to have greater fitness. Large fish are also more valuable for fisheries because there's more profit in catching a large fish, and they're more valuable for marine predators because they have more calorie content. And for a long time, it was assumed that large fish were able to escape ocean predation, but more recent paper by Seitz et al. At the University of Alaska did satellite tagging and actually showed a pretty high predation rate on large Chinook salmon. Most salmon species also show variable age at maturity. Males often have greater variation in age at maturity than females. Females and males can also have different optimal age at maturity, which is interesting. It's often more advantageous for a female to delay maturation because there's a direct effect on fecundity and large body size. Whereas with males, if they can employ a sneaker tactic and reproduce early, then they may be able to escape ocean mortality. But concerningly, many salmon species have shown declines in size and age at maturity. So right now, there's been a few genes that have been associated with age at maturity. VGLL3 was first identified in Atlantic salmon, and there's not evidence so far that has an effect in Pacific salmon. The 6.6 gene was also identified in Atlantic salmon, and more recently has been identified as influencing age at maturity in sockeye and steelhead. And then in Chinook salmon, this was research that I had done and identified Y-chromosome haplotypes, so extended sections of the Y chromosome that influence male age at maturity. So for VGLL3, these plots are from Barson et al., who initially made the discovery that VGLL3 and 6.6 were associated with age at maturity in Atlantic salmon. And there was two alleles. There's an early and a late allele. And you can see on this plot the average age of maturity for a population is highly correlated with the frequency of the late allele. So as the frequency of the late allele increases, the average age at maturity also increases. But the ultimate phenotypes of the individuals also exhibit sex-specific dominance. So in these plots here on the lower right, we have it separated by males and females. Age at maturity is on the y-axis, and then the genotype is on the x-axis. And the size of the circles is the relative proportion within each genotype. And so this dominance is most obvious for the heterozygotes, the LE individuals. For females, most of the LE individuals are mature at either 2 or 3 years of age. Whereas for males, most of those heterozygotes actually mature at age 1, a significant still at age 2, but rarely at age 3. So this same genotype is having different phenotypic effects in females versus males. And this helps resolve some of that conflict of having different optimal age at maturity between males and females. For 6.6, this gene it's on a different chromosome in Atlantic salmon from VGL3, but it has a similar effect. This is this same sort of plot as on the previous page, but with 6.6 instead. And the same pattern of the increasing late allele frequency has increasing age at maturity. And then in steelhead, this is looking at the data a little differently. This is from a recent paper by Waters et al. And this also looked at Chinook and sockeye, and it found that 6.6 also had a significant effect in sockeye as well, but they didn't provide a chart like this. So this is a different way of looking at the data. The age is on the x-axis, and the probability of maturing is on the y-axis. And so you can see for males, there's a pretty significant difference. Individuals with the GG genotype, about half of them will mature at age 1. And in contrast, heterozygotes and individuals with the AA genotype, less than a quarter of them will mature at age 1. So the GG genotype will have a much higher rate of early maturation in steelhead. And then for the Y-chromosome haplotypes, this was work that I did as a postdoc. I have 2 papers out now. One where we found these haplotypes were associated with male agent maturity in Alaska, and one where it was associated with male agent maturity in the Wenatchee River. I've also done a project finding Y-chromosome haplotypes in Puget Sound. It's not published yet. We unfortunately had no age data available for that project. So we don't know if they influence agent maturity, but it's likely given the relationships we've seen in other regions. So this figure on the right is from the Wenatchee River project, and we had hatchery origin individuals and natural origin individuals. And these are all males because females don't have Y chromosomes. We have the different haplotypes broken up in rows, and then we have a histogram of age for each of those haplotypes. So in the natural origin individuals, we have a really strong relationship. The individuals with the early maturation type predominantly mature at age 3, which is a jack in the Wenatchee River. So it's essentially a jacking haplotype. We have 2 different late maturation haplotypes that mostly mature at age 5, but also have some at age 4. And then we have a broad maturation haplotype that is mostly maturing at age 4 and then has a significant proportion at age 3 and 5. And so this is the natural origin individuals. And in contrast, the hatchery origin individuals, for the late maturation and the broad maturation haplotypes, they all have a big shift towards earlier maturation. And so we see a much weaker association due to the hatchery rearing conditions. And this is consistent with previous projects that have shown that hatchery settings tend to shift maturation of individuals towards earlier maturation. And this might actually be why the Waters paper didn't find significant results for some of their species. When looking at VGL3, they pointed out that in Chinook salmon, they mostly had access to hatchery populations, and overall they did not have a relationship between agent maturity and VGL3, but they did have one natural origin population, and there was a significant relationship when that population was considered on its own. So it's possible that some of these results where we've had non-significance, some of these may actually be a false result. There'll need to be follow-up studies to confirm what's really going on. For migration versus residency, this is particularly important for Micus, both because there's a lot of life history variation in the species, but also because management is defined by migration. Only migratory steelhead are ESA listed, not the resident rainbow trout. And this is despite the fact that steelhead and rainbow typically form interbreeding populations. So if you look at neutral markers, you can't tell steelhead and rainbows from the same population apart. But there is an inversion on chromosome 5 in Micus that's been associated with residency versus migration. But it's important to keep in mind that this association is not perfect. And interestingly, there's a lot of steelhead populations that are fixed for the resident form of the chromosome. These figures are from Pierce et al. who was the first one that did the whole genome sequencing on the chromosome, although previous study or on the inversion, although previous studies had identified the chromosome inversion as existing and associated with migration. And in this case, there's two really interesting patterns. One is that there's a sex-specific influence on migration. So this is essentially the probability that individuals had a specific genotype and were migratory. And so with these steelhead, when looking at the females, individuals that had either the migratory form were either homozygous or heterozygous for the migratory form, about 70% of those females migrated. And in contrast, if they were homozygous for the resident form, only a little over 25% migrated. So a pretty big effect in females, while in males there's a much smaller difference for each of the genotypes. Individuals that were homozygous for the migratory form were more predisposed to migrate, but a much smaller amount compared to females. And in steelhead, females have more to gain by migrating than males because they'll get larger, they'll get more fecund, whereas males, even smaller males, can still obtain reproductive success fairly often. And so there's not as much for them to gain by going to the ocean and risking mortality. There's also regional variation in frequency where the migratory allele becomes rare farther north. And this doesn't mean populations aren't migrating. It just means that the migratory form of chromosome 5 isn't present. This suggests that there's something else going on genetically. And to my knowledge, the only paper that has found an association between this inversion and migration in the north was a paper by Arostegui et al. Showing that this inversion was associated with lake migration in Alaska. And then the last example of these types will be run timing. And one of the big advantages of different run timing is that it allows salmon to exploit habitat that's otherwise unavailable. Salmon returning in the spring can utilize high flow rates to access upriver habitat that's otherwise unavailable for fall returning fish. So it lets them access unique habitat, and then it's also beneficial for people who want to fish year round. The spring fish often are also have a higher fat content, which makes them desirable both for human and animal predators. There's a single region of the genome that's been highly associated with early versus late return timing. This was first identified by Hess et al. In steelhead, and then in the next year by Prince et al. For steelhead and Chinook. And there's been a number of several subsequent studies that have confirmed the initial findings and then expanded our understanding of this region. One of the more recent studies by Thompson et al. Was doing whole genome sequencing and looking at this region that contains two genes, GREB1L and ROC1. And this is a region of low recombination, and so alleles tend to be inherited as haplotype blocks. So essentially, all the alleles at this region are inherited in a single unit. And on this figure on the right, that gives a clearer idea of what's going on. The rows are individuals. The columns are 202 markers that were found to be associated on chromosome 28. And then they're color-coded by early alleles as yellow and late run alleles as blue. And you can see for these spring and winter run individuals, which are the bottom half, there's a lot of the early run alleles. And we tend to see the same patterns for all the individuals. There's one pattern where they're pretty much all of the early run alleles. And then there's another pattern where there's specific fall run alleles interspersed in there. And then for the fall run and the late fall run fish, you can see it's predominantly late fall or predominantly late run alleles. And there appears to be essentially two major types of patterns that are being inherited there. What's interesting is when you look at the run timing of these fish, you can see clear differences based on their overall genotype for this region. fish that are homozygous for the early types. Now these fish down here, they return much earlier than fish that are homozygous for the late alleles. And fish that are heterozygous have an intermediate run type. And those heterozygotes can actually facilitate gene flow between the run types. And in the particular populations that were examined in this paper, when you look at the neutral markers, they actually don't show differentiation between the populations. You really see the differentiation when you're looking at these adaptive run timing markers. So once we know that a marker is adaptive, how do we actually bring that into management? And there's two main things that we can actually do with adaptive markers in management. We can do monitoring or manipulation, and there's different risks that are associated with each of these. For monitoring, we can examine the current diversity in populations. We can look to see what the frequency of adaptive alleles are in different geographic regions. We can look to see what the frequencies are based on different life history types, if we're interested in different run timings or in populations that have different maturation ages. We can use this just to give a better idea of what's going on with populations. When we have projects that go on year to year, we can also look for shifts in the frequency of adaptive genes. We could see if there's gradual shifts towards one allele or towards another. We can also, if we have long-term data sets, we can also look to see if there's shifts in frequencies that are correlated with different environmental conditions. For example, if the blob ears tend to select certain alleles, and non-blob ears tend to select others, we would see fluctuations in the allele frequencies. Another thing we can do is we can compare historic and contemporary samples to look for loss of diversity or for admixture of these adaptive genes due to translocations. We can see if current populations have adaptive alleles that were not there in the past because fish have been moved around by people, either for hatchery or for fishing. And there's monitoring adaptive markers it's pretty low risk. The only real risks associated with it are false or oversimplified interpretation. When these markers are first identified, it's important to keep in mind that they're almost always identified in one population in one specific environment. And so it could be that in a different environment or a different genetic background, they may have a different effect. And so we could, if we have a situation where we've identified it and we interpret it just in the context of that original population, but we start examining it in a lot of other populations, we may not be getting the full story. That doesn't necessarily cause a lot of harm to the populations, though. The other thing that can be done is actively manipulating adaptive markers by trying to adjust the frequency of adaptive alleles, either through supplementation or selection at a hatchery. I see sort of two ways or two purposes to this. One would be just to maintain diversity that might otherwise be lost. So you can imagine a situation where a population has say maybe there's a fall run population that has low levels of a spring run allele, and you want to keep that in there. You don't necessarily want to try to favor one allele or the other. But if it was a hatchery population, you could just monitor the broodstock and make sure at least some individuals are making it to the broodstock each year that have the spring run allele. I see that probably as sort of the first way that there would actually be intervention for adaptive markers, is just to maintain diversity if there's a lot of concern about loss of diversity. The other thing that could be done is actually actively trying to change the phenotype of a population. For example, if you had a fall run hatchery population and you wanted to make a spring run population, you could screen the broodstock and try to make them all spring run. You could also try to shift the age composition of a population by introducing early or late run alleles and screening out individuals that have the undesired alleles. but there's a lot of potential pitfalls to these sorts of activities. I think the biggest one is just the unintended consequences. You don't know what you don't know. Most traits are controlled by multiple genes, and focusing on one gene could miss important variation in other genes. And the interaction among genes may be just as important as the variation in single genes. So with these examples that I've listed, there's single genes or single genetic variants that have a big effect on the ultimate phenotype of the fish. But even that big effect is still a minority of the total variation. So the VGLL3 gene it influences about 36% of the variation in the ultimate age of maturity, which means that 64% of the variation is caused by other genes or the environment. So there's a lot of other things that we don't understand yet. Traits don't exist in isolation, so changing one trait will likely impact other traits. And without a good understanding of what's going on, we might actually introduce negative consequences in other traits. And then at a genomic level, selecting on a single gene may introduce selective sweeps. If there was one particular genetic variant that we are interested in and we keep selecting for that and excluding anything else, then that's going to reduce diversity around that particular variant. And there may be other important genes there that we start losing important diversity in and causing maladaptation. Some other considerations for potential pitfalls is that traits that we consider undesirable may actually have higher fitness under current selective regimes. For example, early maturation in salmon has been increasing, and that might be due to selection. And there's actually a lot of evidence that it is due to selection. And so if we tried to shift populations to later maturation, we might actually result in fewer returns due to an increased mortality. And so in some cases, trying to select for one trait may actually end up negating other actions that we're trying to take. And then finally, environments change. Traits that are beneficial now may become deleterious later. It could happen on a shorter scale, like traits that are advantageous during blob years may be a disadvantage in non-blob years and vice versa. But this would argue for maintaining diversity rather than trying to promote some genetic variants over others. So sort of big picture, where do we go from here? One of the obvious steps is identifying more adaptive markers in more species. In salmon, I think a couple key traits going forward for people to identify adaptive markers are going to be disease resistance and thermal tolerance. Both of these traits have a number of studies showing that there is a genetic component to them. But so far, to my knowledge, they've only identified broad genetic regions and not individual genetic variants that have a high association with disease resistance or thermal tolerance, at least in Pacific salmon. It's also important to get a better understanding of how these adaptive variants act in different environments or in different genetic backgrounds. We've already seen from the Y-chromosome haplotypes that the hatchery ring environment has a much different it causes a much different relationship between the genetics and the age of maturity. And so we would expect this to happen in hatchery environments, but it could also happen in natural environments where fish inhabiting just water that is more productive or has different temperatures may also experience different genetic effects or relationships between their gene types and phenotypes. And finally, there will need to be a framework for management that tries to exploit the advantages of the adaptive markers while minimizing the risk of unintended consequences. So now that we've talked about sort of what some of the possibilities are, I wanted to talk about what we can actually do right now at WDFW. So we use GT-seq for most of our genetic monitoring work. This is a technology that lets us genotype hundreds of markers per individual and thousands of individuals at a time. And the nice thing is that this technology is pretty flexible. So we can add adaptive markers as they're discovered. And in the panel that we have for Chinook salmon, we use this for genetic stock identification, but we have 33 runtiming markers on there. And we just finished developing markers for the Y-chromosome haplotypes in Puget Sound. We'll be testing that on a population this next week to see if we can find associations with age at maturity. We also have markers for Alaska and Wenatchee Y-chromosome haplotypes. They're not in our panels now, but they could be added in the future. So for Chinook salmon, we can currently monitor runtime variation and Y-chromosome haplotype variation in specific geographic regions. And again, that's associated with male age at maturity. And in the Wenatchee River, at least, there's one that's specifically associated with jacking. And for MICUS, we have marker panels that currently have runtiming alleles and have the migration chromosome 5 inversion. So we can also look for runtiming variation and migration variation with the caveat that the chromosome 5 inversion has a weaker and regional association with migration. So just the last thing I wanted to talk about was a project that we did this last year in the Yakima River. This was with co-managers at the Yakima Nation Fisheries, and we conducted genetic stock identification on Chinook salmon caught at the Prosser Dam. And it was a really fun project to work on because the main purpose was just genetic stock identification, trying to assign fish caught at the Prosser Dam back to their population of origin. But these runtiming markers gave us additional information on runtiming variation that we didn't know about. So I'm going to go again over the genetics of runtiming real quick just to frame you up for the next slide. So again, we have the GREB1L and ROC1 genes that influence age at maturity on chromosome 28. And our marker panel has 33 genetic variants throughout this region, represented by those little dots. For each of these different variants, we have one allele associated with early migration and one associated with late migration. And this region is inherited as a single block. So these aren't 33 independent markers. And when we look at how they're inherited, we see 4 basic haplotypes, which are just combinations of the alleles of each of these markers. We see one haplotype that is all early alleles. We see one that's all late alleles. And then we see 2 that we call recombinants. So the first half of those markers are early alleles, and then the second half is late alleles. But some of the specific variants differ in early and late alleles between these 2 recombinants. And each fish will have 2 of these different alleles, one for each chromosome copy. So an individual fish could have 2 of the early alleles. It could have an early and a late. It could have an early and a recombinant, a late and a recombinant, a late and a late, any combination of 2 in there. And when we were doing this this project, we were looking, we were doing a new or updating the baseline for our GT-seq technology, as well as genotyping fish caught at the Prosser Dam. So these all of these collections, except for the Prosser Dam, are baseline samples where fish were caught in their respective river. And what we saw is that the populations that were fall runtiming have a very high frequency of the late allele, which is not any surprise. It's pretty expected. And I believe there's only actually one individual that had the early allele in the lower Yakima fall. When we look at the spring populations, what we see is there's also a high frequency of the early allele, which is expected. The next highest is the recombinant 1 allele, which is up to 26%, so pretty common. The R2 is generally rare, But then what was interesting is the only appreciable frequency of the late alleles is showing up in the Cleellum hatchery population. And when we look at the run timing of fish that were caught at Prosser Dam, we first took the fishes caught at Prosser Dam and assigned them to their population of origin. For the Natchez River fish, we found that most of them are either early homozygotes or early and recombinant heterozygotes. And the peak of migration for the fish that were homozygous for the early allele is May 15th. When we look at the fish that assigned to the Upper Yakima Spring, we see we actually have a decent amount of fish that are either homozygous for the late allele or heterozygous for it. And those have a much later run timing, peaking about first week of June, second week of June. And we'd known from previous data that was collected that the Upper Yakima populations had a later runtime in general than the other populations in here. And this is Upper Yakima reporting group, I believe, includes the Cleellum fish as well. But we didn't know is that run timing difference was due to the presence of the late allele in that population, where it was absent from the other populations. When we look at just the individuals that are homozygous for the early run allele, they have the same run timing in both of the populations. So the overall population, the overall later run timing was really because of those individuals with the late alleles. So in conclusion, adaptive markers can give greater information about the populations that we're monitoring, and monitoring the frequencies of these adaptive markers can help protect important genetic variation in these populations by letting us know if there's variation that is likely to be lost. But more research is needed on how the adaptive markers influence phenotype in different environments and different genetic backgrounds before taking actions where we actually try to modify the phenotypes of populations. And so with that, I'd like to thank the WDFW Molecular Genetics Lab and the Yakima Nation's Fisheries for making some of these projects possible. And I will take any questions.
Garrett, thank you so much. That was really an interesting talk, and you made it accessible to non-geneticists. So thank you. Let's see, I unfortunately cannot, Teams is not working on my computer, only on my phone, so I can't see any questions. So if you could please moderate your own question session, I apologize.
Okay. I will try to do that.
Or maybe somebody else could. Sorry. Or people who just want to unmute themselves, whatever works.
Okay, I think I am pulling up the chat. I am not seeing any questions yet.
We have a hand raised. I can see that. So Bob, go ahead.
Okay, I think I'm all right. So the question I have is, how is all this information going to change a distinct population segment, or the ESU kind of designations by the feds, for the Endangered Species Act? Since they used to, NIMS used to say that some of these runs were not distinct, and are we going to have more differentiation in the future?
I would say that's a distinct possibility. There's a lot of people that are looking into that currently, for sure. And if nothing else, there's already groups that are suing for fish to be managed on these specific genes. I know there's at least a lawsuit in California to manage Chinook salmon specifically on these run-timing genes. So I don't know. I mean, my guess is that in the future there will at least need to be some consideration of that. I don't know how it will actually influencing ESA listings, and it's probably going to be a case-by-case basis. But it's something that we'll probably have to deal with more and more down the line. It's kind of a vague answer. It's definitely nothing definitive, but I think it's at some level things are going to change, and we're going to have to manage on the genes. But most of the people that are working on this also recognize it's not necessarily a good thing to manage on a single gene. There's so many things going on. It's certainly appealing, but there's other things that need to be considered as well.
Well, it does take ecology more into account, that's for sure.
Mm-hmm.
Yeah.
Yeah, and I think the run timing is a good example because there's a lot of populations where the fall and the run are just genetically distinct, or the fall and the spring are genetically distinct, and they're easy to tell apart. But then you have the other situations where the fall and the spring can interbreed through these heterozygotes, and for the rest of the genome they're the same, but they're different at these specific runtiming regions, and so it makes it really complicated how you account for all that variation. It's not like there's not really a good one-size-fits-all solution.
Thanks, Garrett. Anybody else? Any questions? Raise your hand, unmute yourself, write them in the chat.
No, I'm not getting anything in the chat.
Okay. Well, I guess we can end 10 minutes early, but Garrett, I just want to thank you again so much for taking the time to talk to us about this really cool work in a accessible manner. And everyone can go try to keep cool for 10 extra minutes this afternoon. And we'll give Garrett another round of applause. And thank you again.
Yeah, thanks for having me.
Okay.
Yeah.